So now onto meiosis. The point of meiosis is to make different cells genetically instead of like mitosis, which is duplicating cells. However, interphase is similar. We start off with 4 chromosomes, one from the mother, and then one from the father, one from the mother, and one from the father. In interphase, though, the cell is going to make a copy and then go through a growth phase, and then that means it's going to get larger, thus duplicating the DNA. It's the result of interphase, so everything happens the same. Each one is going to make a copy of itself and then continuously to duplicate its DNA and grow even larger. As we enter prophase, there's actually going to be 2 divisions. So as you can see now, those homolog chromosomes have actually formed together, and this is called a tetrad. So what we're actually going to see now is the mom chromosome is going to start wrapping up with the dad chromosome. So just to kind of show here how that works, basically it's going to go like this, as best as I can show that. But basically they're going to wrap together with each chromosome, and same with like the bottom. Basically just make like almost like a braid. It's going to wrap together. So they can wrap, and then we can also start like getting rid of some of these top genes. And what we're actually going to do with that is we're going to switch my marker. We're going to start switching, and you're going to start seeing that the dad side, that like the blue, is now going to have like a green gene. And what happens also in prophase 1 is this creates variability, so these genes will actually start switching and moving around. They're wrapped together, and this is going to create different portions of genes. So after the prophase 1 is metaphase 1, and as you can see, I just put the board blank, and I'm going to redraw what could happen in metaphase. So what I have done is I redrew those chromosomes. So as we see here is some different kind of assortments. So these chromosomes can pair up with like mom, dad, or mom, dad. These can also switch, so the mom can go in between the 2 dads, or the dad can go in between the 2 reds, etc. And basically all this is showing is independent assortment, which can change which side the chromosomes are on, and this is important as each side will be drawn to a different pole. So meaning poles is if you had some sort of disease, if these chromosomes are facing this way, then a certain disease will be attracted and you will have that. Now if these were facing this way, the pole that does not have the disease, and it was attracted this way, you would not have it. Now towards the end of metaphase 1, we start to see spindles appear, again from those centromeres, just like that. And in anaphase 1, we will see these spindles sort of disappear. So right now, I'm just going to erase the spindles along with those chromosomes being pulled to the side, and I will redraw those. So basically what will happen is the chromosome will move over here, and these chromosomes will move over here. Same with these ones moving over here, and these ones moving over here. All right, so now I'm going to redraw what this looks like in anaphase 1 after those spindles are gone. So here's anaphase 1 after those spindles have disappeared. So they were all right in the middle. So now the spindles drew them together, and then in anaphase 1, which is being presented right now, those chromosomes will move to either side. Moving into telophase 1, we now see that those chromosomes will be now formed into 2 nuclei. So what this looks like is now we have 2 nuclei here. So after telophase, we normally would see another interphase as in mitosis, but since this is meiosis, we will see prophase 2 happen, which these chromosomes will line up with each other. And what that looks like is this. So you can see that they lined up and they merge into metaphase 2, which were these chromosomes, will now form into the middle. Now we hit anaphase 2, which will basically split these in half and move to the side. And this is represented by looking like this. So here is that anaphase 2. So these chromosomes will actually split apart and move to the top on either side. So as you can see here, you might be able to know what comes next in telophase 2, as there's 2 chromosomes here, 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 and here. So what that means is we now create 4 nuclei. So in telophase 2, this is going to happen into 4 nuclei. Just like that. So let's erase this, we're not confused. All right. So this is now telophase 2, and we have now have 4 nuclei. This is different from mitosis because, as you can see, these chromosomes are different. In mitosis, those chromosomes are the same in order to function properly. But since we are duplicating chromosomes genetically and making different cells, we do see a reduced number of chromosomes in each nuclei, and they are different, as you can tell by the combinations. So for males, each of these 4 nuclei will actually be sperm. So the males actually get 4, while females, 1 of these cells would be used up. These would end up going away. You would not see these anymore, and everything around you would just be the rest of that nucleus. So you have the egg, then you would have a mitochondria, the Golgi apparatus, etc. And that's the end of meiosis.